Assalamu alaikum, my dear students. Today we are going to talk about bacterial cell structure. Before discussing the components of bacterial cells, let's have a recap of the things we have so far talked about. We discussed kingdom Monera, then we talked about the major group of kingdom Monera, I mean eubacteria. We discussed the general characters of eubacteria, and today we will specifically restrict our focus on the bacterial cell structure. The key components that we are going to talk about under this topic is the envelope, then we will talk about slime layer, or you can say capsule, then cell wall, and we will discuss then later on about cell membrane, cytoplasm, the ribosomes which are present inside cytoplasm, inclusion bodies, nuclear material, and under nuclear material we will talk about nucleide first, and then we will talk about the plasmids. Besides these components, there are some other important components of bacterial cells like flagella, we have villi and fimbriae. We will have a brief overview on all of these components one by one. Without consuming much of your time, let us directly move to the glycocalyx or slime layer or capsule. Okay. Glycocalyx, it is the outermost coat of the bacterial cells. It is composed of sugars. That's why this is known as sugar coat. It is a viscous gelatinous polymer and is present external to the cell wall. You will find glycocalyx placed at the outer surface of cell wall. This glycocalyx is composed of polysaccharides including dextran, levain, etc. and polypeptides which are made up of amino acids. Okay. The glycocalyx, it is not produced at the site where it is located, but it is produced inside the cell and then later on it is secreted outside. Depending upon whether both polysaccharides and amino acids or polypeptides are present in the glycocalyx, there are two types of glycocalyx. One is known as capsule and second one is known as slime layer. The capsule is relatively well organized and firmly attached to the cell wall. It is not easily washed off and is composed of both polysaccharides and amino acids. Okay. Once again, I will tell you. The capsule is well organized and is composed of both polysaccharides and amino acids. It is firmly attached and it is not easily washed off. The slime layer, which is the second type, it is relatively diffused, unorganized material and easily washed off. And you have to remember that this slime layer, it does not contain the polypeptides or amino acids. It is only constituted of polysaccharides. Okay. Now the functions of glycocalyx. Glycocalyx, it gives virulence to the bacteria. Virulence means it gives protection against phagocytosis by host cells. Say for example, this is a host cell. Okay. This is a bacterial cell. And this bacterial cell possesses glycocalyx. Okay. The host cell tries to engulf this bacterial cell, but because of the presence of the glycocalyx, the host cell is not able to phagocytosize this cell or engulf this bacterial cell. The second important function of glycocalyx is it prevents desiccation. It prevents loss of water from the bacterial cells because the glycocalyx has a lot of water retention capability. The third function of glycocalyx is it excludes viruses and most hydrophobic materials from entering the bacterial cells. Okay. Glycocalyx is, say this is a glycocalyx layer. It prevents the entry of viruses and other hydrophobic materials into the bacterial cells which can cause damage to the bacterial cell. Okay. One more function of glycocalyx is it helps the bacterial cells to get attached to the surfaces of solid objects and tissues. Now here you can see there is well marked difference between the slime layer and capsule. In the slime layer you can see it is not that much organized, it is diffuse rather and is composed of polysaccharides only, while as capsule is relatively well organized and is composed of both polypeptides as well as polysaccharides. Now the second one is cell wall. We will talk about cell wall. Considering the complexity and the myriad set of functions that cell wall performs in bacterial cells, it is considered as one of the most important components of bacterial cells. Cell wall is regarded as one of the most important component of bacterial cells because it performs a number of functions and its structure is so much complex. Okay. The cell wall is absent in some groups like mycoplasma, okay, and archaea. Archaea means the ancient bacteria. Okay, these two groups of bacterial cells, they do not possess the cell wall. Now, how does it appear like? The cell wall appears as an electron dense granular structure surrounding the cell membrane, and this electron dense granular structure, I mean cell wall in case of bacteria, it is different from plant and fungal cell walls. The cell wall of fungi and plants, it is quite different from the cell wall of bacterial cells. Now the function of cell wall, it has many functions, and the first one is it primarily functions as a mechanical agent. It gives shape to the cell. Okay, say for example this is a bacterial cell. Without cell wall it will not have any specific shape. The cell wall gives it a permanent shape. Okay, that means it gives mechanical structure to the cell, bacterial cell. Second one is it protects the bacterial cell from osmotic lysis. Say for example you have placed a bacterial cell in a medium and that medium is hypotonic medium. Hypotonic means less concentrated. The concentration of salts inside bacterial cell is more as compared to the concentration of salts in the medium. Obviously water will tend to move inside the bacterial cell, and if this continues unabated, that means water continues to move into the bacterial cell, it will ultimately burst. The cell will burst. Okay, the cell wall which is present on the outer surface of cell membrane, it tends to resist, it tends to prevent the further expansion, it tends to prevent the further movement of water into the cell. Okay, देखो ये जो cell membrane है, it will tend to move when water enters the cell, but the cell wall applies the opposing pressure. Okay, ये अंदर की ओर प्रेशर डाल रहा है so that the plasma membrane cannot move outside. In nutshell, you can say it gives protection against osmotic lysis. One more function of cell wall is it grants pathogenicity to the bacterial cells. The cell walls of many bacterial cells contain, it has been found that the cell walls of many bacterial cells contain the antigenic components. Okay, antigenic components are those specific molecules which give their bacteria or other pathogen their pathogenic behavior. Okay, say this is the cell wall of bacterial cell. Okay, this is the cell wall. It contains some molecules, and on the basis of those molecules, this bacteria is able to act as a pathogen. Okay, so you can say the cell wall, it functions, it gives pathogenicity to the bacterial cells. One more function of cell wall is it provides protection from some toxic substances. Okay, it provides protection from toxic substances. Due to this fact, a number of antibiotics have been developed. Okay, a number of antibiotics have been developed which specifically target the cell wall of bacterial cells. You know that bacterial cell wall is giving it protection against osmotic lysis. It is giving them protection against toxic substances. We wish to kill the bacterial cells with the help of some toxic substances, but because of the presence of cell wall, the toxic substance cannot grant an entry into the bacterial cell. And considering this, a number of antibiotic substances have been produced which specifically break down the cell wall of the bacterial cells. Once cell wall is broken down, there is nothing to prevent the entry of toxic substance, and as such the bacterial cell gets destroyed. I think this is all for today. In the next class we will continue to discuss about the cell wall of bacteria. We will discuss its composition, then we will talk about gram positive and gram negative bacteria. then the procedure of gram staining, and many other things. Uh, I think we should call it a day. Thank you and stay blessed.